Welcome to the Deep Dive. Today we're taking a really vital journey into the world of pharmacologic control, specifically how we use antimicrobial drugs to fight infection, and maybe more importantly, why this incredible success story is, well, rapidly becoming one of the biggest challenges in modern healthcare.
It really is.
If you look back, the timeline for treating infectious diseases,
Yeah.
the discovery of these drugs is actually astonishingly recent. You had Pasteur and Joubert noticing some microbial conflict way back in 1877, sure. But the big one everyone knows is Fleming, right? Alexander Fleming and that famous mold penicillin in 1929.
Mm-hmm. And we often focus on those, natural discoveries, which were groundbreaking, but the synthetic revolution was just as critical. Gerhard Domagk's work in 1935 with Prontosil, that gave us the first effective synthetic sulfonamides. It showed we could actually engineer molecules to kill germs. That really set us on the path we're still following today.
Okay, so let's establish the mission here. The core goal, right, for every single antimicrobial agent, doesn't matter if it's fighting bacteria, viruses, fungi. The idea is always to interfere with the pathogen, messing with its metabolism, its structure, basically stopping it so profoundly it can't survive or reproduce. That's the fundamental aim.
Exactly. And for you, the learner heading into healthcare, the very first distinction you absolutely need to get is the action. How does the drug affect the microbe? We classify them basically two ways. They're either microbicidal or bactericidal, meaning they kill the organism flat out, or they're microbiostatic or bacteriostatic. And that means they reversibly inhibit growth. They stop it, but don't necessarily kill it.
Ah, okay. So if we only inhibit the growth, we're essentially outsourcing the final blow, the kill, to the patient's own immune system.
Precisely. If a drug is just static, the host's own defenses have to step up and clear that infection, which means, understanding the patient's underlying health is absolutely crucial. If someone's immunocompromised, a static drug might just not be enough. So this difference killing versus just stalling, it's foundational. It really dictates the strategy. And interestingly, no matter what the target is, bacteria, virus, whatever, all these agents attack through about 5 basic categories of mechanisms.
Okay, let's dig into those. The first strategy, it seems almost elegant in how it targets things.
Yeah, that would be inhibiting cell wall synthesis. This is often the easiest target, especially in bacteria, because their wall is made of peptidoglycan. Human cells just don't have that. So drugs like penicillin, cephalosporins, they jump in during that final assembly stage. They prevent the crucial cross-linking that gives the wall its strength, its rigidity.
And the result is just catastrophic failure for the cell.
Oh, absolutely. The wall weakens, especially at points where the cell is growing. And then if that bacterium is in a hypotonic environment, think like normal body fluids, water rushes in, internal pressure builds up, and boom, the cell lyses, it bursts.
Wow.
It's highly effective and, like you said, beautifully selective because we don't have that target.
Okay, so if breaking the wall isn't an option, the next best thing seems to be crippling the factory inside. Inhibiting protein synthesis. How do we attack the bacteria's manufacturing hub, the ribosome, without messing up the patient's own cells?
This is where selective toxicity gets really, really interesting. See, bacterial ribosomes are what we call prokaryotic 70S ribosomes. Structurally, they're distinct from our own eukaryotic 80S ribosomes. And that difference, that's the molecular Achilles heel we exploit.
Gotcha.
So drugs like tetracyclines, macrolides, chloramphenicol, they can target that 70S structure in a few ways. They might prevent synthesis from even starting, or block the peptide bonds from forming, or just stop the ribosome from moving along the mRNA strand to read the message. All effective ways to shut down production.
That structural difference, it's really a gift for drug design, isn't it?
It absolutely is.
Okay, moving on to mechanism 3, inhibiting nucleic acid synthesis. This sounds fundamental, stopping DNA and RNA from being made, so stopping replication and transcription. Lots of ways to attack this, I imagine, but the text highlights one really ingenious strategy, competitive inhibition.
Yes. This strategy is just a masterclass in exploiting a metabolic weakness specific to prokaryotes. We use agents like sulfonamides and trimethoprim. What they do is molecular mimicry. They look almost exactly like the normal substrate that a crucial enzyme needs. So they block that enzyme, preventing the cell from making essential precursors, specifically, something called tetrahydrofolic acid. And bacteria need that acid to make their nucleic acids.
So the drug basically acts like a fake key jamming the lock. But why is that selectively toxic? Why doesn't it harm us?
Because it exploits a fundamental difference in how we get our nutrients. Bacteria must synthesize their own folic acid to survive. They have to make it. Humans, on the other hand, We get all our folic acid through our diet. We don't synthesize it. So if we block that synthesis pathway, we only harm the bacteria. The host isn't affected because we get ours pre-made. It's perfect targeted drug design.
Brilliant. Okay, next, mechanism 4. Disruption of the plasma membrane. Now, every living cell needs a membrane, right? So disrupting it seems like a pretty direct way to cause damage.
Oh, it causes catastrophic damage. It hits the cell's physical integrity, messes up all the transport functions. It's bad news for the cell. And we have agents like polymyxin B that target bacterial membranes and antifungals like, say, amphotericin B that go after specific lipids in fungal membranes. And this is a really critical caveat for any healthcare professional because all cells, including our own human cells, share similar membrane components. These types of drugs often carry a higher risk of toxicity to the patient. You have to be much more careful with them.
That makes sense. There's less difference to exploit there.
Yeah.
And finally, mechanism 5. This is a bit broader. Inhibition of metabolic pathways. Basically anything that blocks key enzyme activity outside of making DNA or proteins. Often involves essential compounds, like NAD, the text mentions. Seems like a catch-all category, but it loops back.
It does. It loops right back to our competitive inhibitors, the sulfonamides and trimethoprim. They actually fit here too, because by disrupting the synthesis of that tetrahydrofolic acid precursor, they're effectively inhibiting a crucial metabolic pathway beyond just direct nucleic acid synthesis.
Okay, so now we know how they work, those 5 main ways. The next massive clinical question is, which drug do we actually pick?
Right, and that selection process starts with that principle we keep mentioning, selective toxicity. You want to kill the pathogen, spare the host. That's goal number 1. And right alongside that is the therapeutic index.
Explain that concept for us, the therapeutic index.
Sure, think of it as a safety measure. It's basically the ratio between the toxic dosage, the amount that starts harming the host, and the therapeutic dosage, the amount needed to actually eliminate the pathogen. The wider that range, that gap between effective dose and harmful dose, the better and safer the drug generally is.
So a narrow index means you're walking a tightrope.
Exactly. A narrow index means the dose that works is very close to the dose that causes harm. You need much more careful monitoring with those drugs.
Okay. And then we categorize drugs based on their spectrum of action. We hear these terms a lot. First, narrow spectrum drugs, like penicillin G, the example given, effective against only a pretty small number of microbes.
Yep. And the big advantage of narrow spectrum is you minimize the collateral damage. You're less likely to wipe out the patient's beneficial normal flora, which in turn reduces the risk of a secondary infection popping up.
Okay, then the opposite end. Broad spectrum drugs, like sulfa drugs. These hit a huge variety, gram-positive, gram-negative, lots of different bugs. But it always feels like a bit of a gamble, doesn't it? Kind of a scorched earth approach. Wiping out the body's natural defenses, just hoping to hit the right pathogen.
You've absolutely hit the nail on the head there. That is the risk. The major disadvantage is exactly that. Destroying beneficial normal flora, that can lead directly to what we call a superinfection. Basically, resistant organisms that weren't bothered by the drug suddenly have no competition. They're given free rein to overgrow in that now empty ecological niche.
Nasty stuff.
Very. And then there's medium spectrum in between, effective against some, but definitely not all, of the major gram types.
Beyond the spectrum, though, there are practicalities, like can the drug even get to where the infection is?
Oh, absolutely critical. Delivery is key. The drug has to reach the site of infection in an effective concentration. Prime example, CNS infections, like meningitis. The drug has to be lipid-soluble to cross the blood-brain barrier. If it can't cross, and many non-lipid-soluble agents can't, it's basically useless for that infection. That's why we rely on drugs like certain third-generation cephalosporins for those deep brain infections. They can get in.
Okay, so you've selected a potential drug, considered its index, its spectrum, whether it can reach the target. Now you have to know if it will actually work against the specific bug this specific patient has. We can't just guess. What are the standard lab tests for confirming effectiveness?
Right. We rely on 3 main methods. The first one, probably the most common visually, is the disc diffusion test, also called the Kirby-Bauer test. You basically spread the patient's pathogen all over an agar plate, then you place little paper discs soaked with different antibiotics onto the agar, you incubate it and wait. If the drug works against that bug, you'll see a clear ring around the disc where nothing grew. That's the zone of inhibition.
Okay, but seeing that clear zone, that doesn't necessarily mean the drug killed the bacteria, does it? It just shows growth was stopped. It could be static.
Exactly right. The size of the zone is useful, gives you a clue, but it doesn't guarantee the drug is microbicidal. It just shows inhibition. To get a more precise measure, especially of how much drug is needed, we use the dilution method to find the minimal inhibitory concentration, or MIC. For this, you take the pathogen and test it in a series of tubes, each with a decreasing concentration of the drug. You look for turbidity cloudiness, which indicates growth. The MIC is simply the lowest concentration of the drug in a tube that shows no visible growth. The tube stays clear.
Okay, that tells you the minimum needed to stop growth.
Correct. And then, if the clinical situation really demands knowing if you killed the organism, not just stopped it, you take a small sample from that clear MIC probe, and you put it onto a fresh agar plate without any drug. This is called subculturing. If nothing grows on that new plate, it means the drug in the original tube actually killed the bacteria. You've now determined the minimal bactericidal concentration, or MBC.
MIC stops it, MBC kills it. Got it.
And one more, briefly, the serum killing power test. This usually lets us test the patient's own blood serum, which contains the drug they're taking against their specific pathogen. It gives a sense of effectiveness in vivo inside the body.
Okay, very important tests. But we absolutely have to pause here and talk about the downsides, the risks. Antimicrobials are miracle drugs, yes, but we know they carry significant side effects. The text mentions worries about hepatotoxic effects on the liver, nephrotoxic on the kidneys, and even damage to the nervous system or bone marrow. These aren't trivial concerns.
Not at all.
And looping back to superinfections, you mentioned C. diff earlier. These aren't just minor annoyances. When a broad-spectrum drug wipes out your normal gut flora, it really does open the door for resistant bugs to take over. Things like Clostridium difficile causing severe, sometimes life-threatening colitis, or fungi like Candida albicans causing thrush or yeast infections. It happens all the time.
It's a constant, complex balancing act. Consider tetracycline. It's usually contraindicated in young children because it can cause permanent discoloration of their tooth enamel. Or certain aminoglycosides, which are known to potentially cause nerve damage leading to deafness or dizziness, serious stuff. But on the flip side, pharmacologically, we also look for positives, like synergism, where using two drugs together makes them way more effective than either one alone, exponentially better sometimes. But we have to strictly avoid antagonism, where combining drugs actually makes them less effective. They interfere with each other.
Okay, synergism good, antagonism bad.
Basically, yes. And all of this, the mechanisms, the selection, the side effects, it all brings us crashing into the biggest, most urgent issue facing global healthcare right now: antimicrobial resistance, AMR. We are quite literally fighting an evolutionary arms race that is being driven by our own success with these drugs.
And this resistance,
yeah,
it's acquired through genetic change, right? Either a random spontaneous mutation happens, or, much more worryingly, microbes can acquire resistance genes from each other on these things called R plasmids.
Yes, R plasmids are like the genetic superhighway for resistance. They're pieces of non-chromosomal DNA floating around, and they can carry genes for resistance to multiple drugs. And they can be transferred horizontally, meaning between different bacteria, even different species, through processes like conjugation, transformation, transduction.
Wow.
This is why resistance spreads so incredibly fast and can jump between different types of bacteria. A problem in one place quickly becomes a global threat.
What's kind of counterintuitive, or fascinating maybe, is that the antibiotic itself doesn't cause the mutation.
Right.
It doesn't make the bug resistant.
Exactly. That's a common misconception. The drug creates intense selective pressure. Think of it like this. In a huge population of bacteria, maybe one or two, just by random chance, already have a mutation that makes them slightly resistant. When you introduce the antibiotic, it kills off all the sensitive ones. Suddenly, those few resistant mutants have no competition and a wide open field. They survive, they multiply. Especially if the drug is misused, not taken long enough, wrong dose, used for viruses. We're basically just selecting for the superbugs. We're breeding them.
That's a chilling thought. We're driving their evolution.
We are.
So let's detail the ways the microbes fight back, their counter-strategies. The text lists 6 main mechanisms of resistance. First, they can just change their membrane permeability, basically modify the door so the drug can't get in.
Right. Or second, they can develop pumps for increased drug elimination. These are specialized proteins, sometimes called multidrug-resistant pumps, that actively grab the drug inside the cell and pump it right back out before it can do anything.
Clever. Okay, third, they can change the target or the receptor. So if the drug normally binds to a specific spot on the ribosome, for example, the microbe alters that spot so the drug just can't attach anymore.
Precisely. Fourth, they can sometimes change a metabolic pathway. If a drug blocks step B in a pathway from A to C, the microbe might evolve a totally different route to get from A to C, bypassing the block.
Finding a detour.
Finding a detour, exactly. Fifth, similarly, they can change a previously inhibited enzyme. So the drug used to inhibit enzyme X, but the enzyme mutates slightly, so the drug no longer fits or blocks its action, even though the enzyme still works for the cell.
And finally, maybe the most famous one, developing defensive enzymes. The classic example is B-lactamase, which lots of bacteria now have.
Also known as penicillinase. These enzymes physically find the antibiotic molecule, like penicillin or its relatives, and they break a key part of its structure, the beta-lactam ring. They destroy the weapon itself.
Incredible defense mechanisms.
When a pathogen picks up defenses against 3 or more different types or classes of antimicrobial agents, we call them multiple antimicrobial resistant strains, or MAR strains. These are the infamous superbugs. And you find them concentrated in healthcare settings, hospitals, nursing homes, because that's where the selective pressure from antibiotic use is most intense.
And MRSA, methicillin-resistant Staphylococcus aureus, is the poster child, really. It highlights another issue, too, cross-resistance.
Yes, often resistance to one drug in a class, like methicillin, quickly leads to resistance to other similar drugs in that same class, even if the bug hasn't seen them before.
This is sobering. So the call to action, then, for everyone listening who's going into or is in healthcare, it has to be urgent. What are the key strategies to try and control this AMR crisis?
Number 1, absolutely fundamental. Healthcare professionals must practice strict hygiene. Constant, consistent hand washing. Using isolation procedures correctly for patients with known MAR infections. It sounds basic, but it's critical. Second, physicians need to be careful stewards of these drugs. Use narrow-spectrum agents whenever possible once the pathogen is identified. And absolutely never prescribe antibiotics for viral infections where they have no effect.
And for us, as patients, what's our role?
Hugely important. You must complete the full course of treatment exactly as prescribed, even if you start feeling better halfway through. Stopping early just encourages resistant survivors. And never, ever take leftover antibiotics or medication prescribed for someone else. That's incredibly dangerous.
Right.
And then, of course, there's the larger societal issue, the ongoing debate about the extensive overuse of antibiotics in livestock feed, which contributes massively to the environmental pool of resistance genes.
Okay. Before we wrap this deep dive, we should briefly touch on the rest of the antimicrobial toolbox, the non-bacterial agents. Because killing bacteria is hard enough, but targeting fungi, protozoans, helminths, worms, that presents unique challenges, doesn't it? Because they're all eukaryotes, like our own cells.
Exactly. Selective toxicity becomes much, much trickier.
So, starting with antiviral agents. Here, the drug has to target some part of the viral life cycle, but without destroying the host cell the virus is hiding inside. We see clever strategies, like acyclovir, which messes with viral DNA replication by pretending to be guanine, a DNA building block, or AZT for HIV, inhibiting the crucial reverse transcriptase enzyme.
Right. But a key limitation you have to remember, antivirals generally cannot affect viruses once they go into a latent phase, just hiding dormant in our cells. They only work on actively replicating viruses.
Okay. Then antifungal agents. Again, eukaryotes, so finding unique targets is hard. What do we typically go after?
Often we target structures that are unique or at least different in the fungal cell. A common target is the sterols in the fungal cell membrane or gastral, mainly which is different from human cholesterol, and proterosin B works this way. Or newer agents like the echinocandins target specific components needed to synthesize the fungal cell wall, which human cells lack entirely.
And finally, the really tough ones. Antiprotozoan and antihelminthic agents, drugs against parasites like amoebas or malaria, and against worms. Again, eukaryotes.
Yeah, very challenging. Mechanisms here often involve things like disrupting the parasite's DNA, metronidazole does this, or sometimes physically paralyzing the worm like piperazine or ivermectin, or blocking essential nutrient uptake, like how mebendazole stops worms from absorbing glucose. It's a diverse group of strategies for diverse parasites.
It really covers a vast landscape of microbiology and pharmacology.
It does, and hopefully this deep dive into pharmacologic control really reinforces those essential concepts you need to carry forward. Remember the 5 basic mechanisms of action, they dictate treatment strategy. Remember that selection has to carefully balance the spectrum of activity against that crucial therapeutic index. And know that clinical decisions really rely on accurate efficacy testing using methods like the MIC and sometimes MBC to guide therapy. Ultimately, the challenge facing every single healthcare professional today isn't just treating an infection. It's fighting the relentless, ongoing evolution of resistance. An evolution driven, ironically, by the very success of these life-saving tools we rely on.
That puts it perfectly, which leads to the final thought we want to leave you with today. Given this rising tide of resistance and the worrying fact that the pipeline for discovering truly new classes of antibiotics has slowed, how do you think this constant, intense selective pressure will shape the next generation of infectious disease treatments? And maybe more critically, what new non-pharmacological strategies, perhaps things like phage therapy or much stronger preventative public health measures, will healthcare be forced to adopt, maybe sooner than we think? Something to consider. That's all for this deep dive. Thank you so much for tuning in and learning with us from the Last Minute Lecture team. We appreciate you listening.